bioeducator.com. In today's lesson, we're going to continue on with our discussion of the cell cycle, focusing now on mitosis. Before we do that, let's look at where mitosis is placed in the cell cycle. Recall that there are two major phases of the cell cycle. Interphase, in which the cell spends about 90% of its time, and the M phase, in which the cell spends 10% of its time. Interphase consists of the G1, S, and G2 phases. Recall that the G1 phase is also called the gap 1 phase, and this is a growth phase. The cell is duplicating its organelles, so it's making new organelles, cytoplasm, protein, cell membrane components, everything the cell will need to split into two daughter cells. S phase is the synthesis phase. This is the phase in which the DNA is replicated, so DNA replication. Because if the cell is going to create two daughter cells, there needs to be enough DNA for the daughter cells. During the S phase, growth is continuing, so there is still production of new organelles and cell membrane components going on during the S phase while synthesis of DNA is going on. Synthesis of DNA will be covered in detail under the molecular biology lecture. G2 is gap 2. This is another growth phase. Gap 2 phase, continued growth of the cell. This is all in preparation for the division of the cell into two cells, two daughter cells. M phase consists of mitosis and cytokinesis, so we're focusing on mitosis today. And what mitosis is the separation of the chromosomes into two sets, one for each daughter cell. Cytokinesis is the actual division of the parent cell into two daughter cells. So everything's been duplicated. The chromosomes are split into two sets, one for each nuclei. And then during cytokinesis, the cell pinches off in the middle to create two physically separate daughter cells. Recall also from the cell cycle lecture that extremely specialized cells like neurons don't divide. So they exist instead in a quiescent phase called G0. These are called post-mitotic cells. G0 is sometimes placed here in the cell cycle. Other people, other scientists consider it just an extension of G1, and they'll show it somewhere in here. Either way, it's just an arrested phase or resting state, and these type of cells, like neurons, don't proceed on through the cell cycle. All right, now we're going to go ahead and focus on what happens in mitosis. Let's begin with just an overview. Mitosis is a separation of chromosomes into two sets, one for each of the two identical daughter cells that result. And it's important to understand chromosome structure, so if you need a review on that, that's covered in the cell cycle lecture. But just briefly, prior to the S phase, let's say the cell is in G1. The chromosome will exist in such a way that it has only a single chromatid. After S phase, or during S phase actually, the chromosomes, the DNA is all duplicated. It's exactly replicated. And what this is going to create is a chromosome that has two sister chromatids. And these are identical, sister chromatids. This is considered one chromosome. This is still one chromosome. It has two chromatids, but it's still a single chromosome. So don't let it throw you off that this looks different than this, because this is just duplicated information right here. All right, so what happens is the cell is, this cell, let's look at this cell right here, has 1, 2, 3, 4 chromosomes. It goes through mitosis. After mitosis, the result will be two identical daughter cells. But what you'll notice is that those daughter cells don't have sister chromatids. So S phase has occurred. There's been a duplication of this DNA so that one sister chromatid from each chromosome goes into each of the daughter cells. No information is lost that way. There are 4 chromosomes here. There are 4 chromosomes here. The large purple one, that information's here. The small purple one, here. Large green here, small green here. Now, what do these numbers mean? 2n here and 2n here. Well, there are 2 different types of cells that exist in terms of chromosome number. Diploid cells have 2 sets of chromosomes. Haploid cells have only 1 set of chromosome. In mitosis, chromosome number is conserved. So here, these 2 chromosomes are similar in their length and their placement of the centromere. In addition, let's say that these were stained in the laboratory, and you found that they had the same staining pattern, banding pattern. Different bands would form on different locations. And if the length is the same, the centromere location is the same, the banding pattern's the same, we call those homologous chromosomes. So homologous chromosomes carry genes for the same traits on them. So I'm going to call this chromosome number 1, and these are chromosome number 2. In this individual, she received 1 chromosome 1 from her mother and 1 chromosome 1 from her father. She received 1 chromosome 2 from her mother and 1 chromosome 2 from her father. So you look at any somatic or regular body cell, not a reproductive cell. All somatic cells are going to have 2 sets of chromosomes in them, and they are therefore diploid. So these somatic cells are diploid. Homologous chromosomes, chromosomes that are the same length, have the same centromere placement, same banding pattern, those are more superficial things. The important thing is that they carry DNA for the same traits. In other words, let's say chromosome 1 is the chromosome that codes for eye color. Then this individual received a gene or a form of a gene, which is called an allele. Allele are different forms. Alleles are forms of a gene, alternative forms of a gene. So let's say chromosome 1 encodes eye color. And this individual inherited the blue eye allele from her mother. So there's DNA here that encodes for blue eye color. Maybe this individual inherited the allele or form of the gene for brown eye color from her father. So these homologous chromosomes carry information about particular traits in alternative forms. Maybe the gene for height is on chromosome 2, and this individual inherited shorter stature from her mother and taller stature gene from her father. So cells that are diploid contain 2 complete sets of chromosomes. What's really important in mitosis is that this chromosome number is conserved. The daughter cells are identical in terms of the information they contain to each other and to the parent cell, and they are still diploid. So mitosis, you start out with a diploid cell, you end up with 2 diploid daughter cells. Now I have 2n written here. Diploid cells, in terms of chromosome number, are known as 2n. Haploid cells are n. Now, in humans, we have 46 chromosomes. Different species may have different numbers of chromosomes. For example, cats, the diploid cells in cats have 38 chromosomes. So humans, our somatic cells, you took out a skin cell and looked at it, you would see 46 chromosomes. And this exists in 2 sets of 23 pairs of chromosomes. That's the easiest way to look at it, that this is 23 pairs. Chromosome 1, chromosome 2, there's a pair. Chromosome 3, there's a pair. Chromosome 4, and all the way. So somatic cells are diploid. They contain 23 pairs of chromosomes. One thing to note is that the 22 of the chromosomes are just numbered. They're numbered 1 through 22. And these are called autosomes. The 23rd set is the sex chromosomes. We will definitely get into this later on when we talk about reproduction, but just briefly, remember that XX, that chromosome conformation is for a female. XY is male. So there are what we call autosomes, chromosomes 1 through 22, and you will have 2 pairs of those. You'll have 2 chromosome 1s, 2 chromosome 10s, 2 chromosome 20s, 1 from your mother, 1 from your father. In addition, you have a set of sex chromosomes. Females have XX, males have X and a Y. All right, so in mitosis, you start out with a diploid cell, 2 full sets of chromosomes, you end up with a diploid cell. We're going to talk about meiosis separately, but just keep in mind that in meiosis, you start out with a diploid cell, but you end up with a haploid cell. Now, delving into how mitosis works. To understand mitosis, you should have an understanding of the structure of the spindle apparatus. We touched upon this in the earlier lecture on cell structure in a discussion about motility within the cell and microtubules. Focusing here on the spindle apparatus. This consists of centrosomes, microtubules, and asters. These are sometimes called spindle microtubules or spindle fibers. And what the spindle apparatus does is it physically separates the chromosomes into 2 sets. In mitosis, it pulls apart sister chromatids so that there will be one set on one side of the cell, the other set on the other side. Let's look at each part of the spindle apparatus, starting with the centrosome. So the centrosome is right here, and within the centrosome, these 2 brown structures are actually centrioles. There are 2 centrioles located within each centrosome, and these are microtubule organizing centers. They're a type of microtubule organizing center. Short version of this is MTOC. And this title tells you what they do. They organize microtubules. These spindle fibers here, or spindle microtubules, are organized right here at the centrosome. So centrosomes are a type of MTOC, and within those exist centrioles. Spindle fibers or spindle microtubules connect the centrosome to the chromosome. Spindle microtubules connect at a specific part on the chromosome called the kinetochore. So kinetochores are located on the centromere, and each chromosome has 2. So let's use red. Here is kinetochore. There are 2 of them, and the reason there's 2 of them is the goal in mitosis is to separate the sister chromatids, and that can only occur if this one pole of the cell is over here, one pole is over here, and the spindle fibers on this side are connecting to the kinetochore and this sister chromatid. The spindle fibers radiating out from this side are connecting to the kinetochore on the other sister chromatid, so that when these pull, when they shorten up, they will pull apart the sister chromatids. Recall from the cell structure lecture that microtubules can add subunits to one end and remove them from an end. Microtubules are formed from subunits of tubulin protein. So these tubulin subunits can be added on to make the microtubule longer so that it can connect to the kinetochore, attach to the kinetochore, and then they can remove subunits to become shorter to pull apart the sister chromatids. So that's how microtubules allow for motility. All right, so we covered centrosomes. Within them are centrioles. These are microtubules, and now asters. What asters are, if you look right here near the centrosome, this area of microtubules that's radiating out, some people consider this a star shape, and that's where it gets that name aster. So these microtubules that are just radiating out from the centrosome are called asters. This is really the situation for animal cells. Plant cells and fungi do not have centrosomes, but they still have microtubule organizing centers. So centrosomes are one type of MTOC, but there are other types, and plants and fungi do have microtubule organizing centers. They still go through mitosis, and they have organized microtubules, but the control is not via a centrosome. It's some other type of structure. Now that you understand the spindle apparatus, it's time to go on to the next, to the first phase, and to focus on the first phase of mitosis. The first phase is called prophase. So mitosis consists of prophase, metaphase, anaphase, and telophase. You should know the major occurrences for each phase. So starting with prophase, during prophase, the nuclear membrane begins to break down and the nucleoli disappear. So in order to separate these chromosomes out into two groups for the two daughter nuclei, the nuclear membrane needs to be removed. So this is shown as a dotted line because the nuclear membrane is breaking down. The last cell I showed you to illustrate the spindle apparatus already had a spindle apparatus completely in place, but the spindle apparatus doesn't exist throughout the whole cell cycle. It has to be formed. So the spindle apparatus starts to form during prophase. And here you can see the centrioles within the centrosome, and the aster, so the spindle fibers, are starting to radiate out. So here we have formation of spindle apparatus. In addition, the chromatin condenses. In the previous lecture discussing the chromosome structure, I mentioned that chromosomes consist of chromatin. Chromatin is DNA plus protein. And the type of protein is histone protein. So the DNA is wound around histone proteins. And for most of the cell cycle, the chromatin is in a very non-condensed state. And if you looked at a cell in most of the cell cycle, you would not be able to visualize chromosomes via light microscope because they're in their uncondensed form. Now, at prophase, the chromosomes condense. The chromatin condenses and forms what most people recognize as, okay, that looks like a chromosome. What we're picturing is the state that the chromosomes are in during mitosis, which is their condensed form. And at this point, the chromosomes would be visible via light microscopy. So chromatin condenses. And the chromosomes, now they form the form of chromosomes that is visible via light microscopy. So three major events: formation of the spindle apparatus, breakdown of the nuclear membrane, and the chromatin condenses. One other thing happens. The nucleoli disappear. So there were some nucleoli visible in the nucleus, but now they disappear. Okay, so that is prophase. The second phase is metaphase, second phase of mitosis. During the metaphase part of mitosis, the chromosomes line up single file along the metaphase plate. And this single file is very important because it's one thing that differentiates mitosis and meiosis. In meiosis, chromosomes line up differently, and that makes a big difference in the outcome of that type of cell division. So here, the cell, keep in mind, has 1, 2, 3, 4 chromosomes, and these are 2 sets of 2. These 2 chromosomes are homologous chromosomes. I'll call them chromosome 1. One from this individual's mother, one from his father. Chromosome 2 from his mother, chromosome 2 from his father. So I have 2 sets of 2 chromosomes, and this cell is diploid. Now, these chromosomes line up single file along what's called the metaphase plate. And all the metaphase plate is an imaginary plane equidistant from the 2 centrosomes. So imaginary plane equidistant from the centrosome. And one other term you'll hear is the pole of the cell. So this is one pole of the cell, one end, and this is the other. And this centrosome is located at one pole. And remember that these spindle fibers are attaching to the kinetochore on one sister chromatid. The spindle fibers on the opposite pole are attached to the kinetochore on the other chromatid, all the way down. So we have, it's going from centrosome to spindle fiber to kinetochore. In this drawing, the sister chromatids are shown for clarity as having a little bit of space between them, just so you can differentiate that there are 2 sister chromatids. But in reality, they're actually held pretty tightly together by proteins called cohesins. Okay, so cohesins are proteins that hold sister chromatids together. These have to be cleaved so that the 2 chromatids can separate, but generally sister chromatids are held together via cohesins. So we did prophase and metaphase. Next phase is anaphase. This is the time when the 2, so we had a chromosome that had 2 sister chromatids held together. Now what's going to happen is the spindle fibers will shorten up. These are microtubules, so they're capable of becoming longer or shorter. The cohesins, therefore, will have to be cleaved. So the cohesins are cleaved, the sister chromatids separate and are pulled to opposite poles of the cell. Remember, I started out with 1, 2, 3, 4 chromosomes, and I can see I still have 1, 2, 3, 4 chromosomes. No information was lost because each chromosome had been through S phase, synthesis phase, and the DNA had been replicated. So if this right here was information encoding for blue eyes, that blue eye information is going to go into this daughter cell. This blue eye information is going to this daughter cell. If this was brown eye allele, this is going into one cell, this is going into the other. A gene for a particular height, an allele for another height. All the information is here. Blue eyes, brown eyes, tall, short. Blue eyes, brown eyes, tall, short. So no information is being lost. Remember that also during the growth phases, G1, synthesis phase as well, and G2, organelles, cell membrane, cytoplasm, cell components were all copied, more than were made. The cell increased in volume. So you can see that the daughter cells will each have everything they need. Finally, telophase and cytokinesis. I put these together because cytokinesis actually begins in late anaphase. It overlaps also with telophase and then continues on after. It's not a clear telophase ends and then cytokinesis begins, so I put these together. During telophase, some of what happens is a reverse of prophase. During prophase, the nuclear membrane broke down. Now the nuclear membrane is reforming, except now there's two nuclear membranes because there's two nuclei, one for each daughter cell. Chromosomes become less condensed. I mentioned that during prophase, the chromosomes condense, the chromatin condenses, and the chromosomes become visible under light microscopy. Now the chromosomes are going to decondense, and you're not going to be able to see them with a light microscope anymore. Nucleoli reappear. They disappeared during prophase, they're reappearing now. So we have nuclear membrane reforming, chromosomes becoming less condensed, the nucleoli reappear, and the other thing, notice, is that the spindle apparatus is going to break down. It formed in prophase, it's going to break down now. It's not needed anymore. Cytokinesis began during anaphase, and cytokinesis is the division of the cell into two physically separate cells by dividing the cytoplasm. In animal cells, this occurs by the formation of a cleavage furrow. This is the cleavage furrow. It's an indentation or a groove in this parent cell. Actin and myosin will pinch off this parent cell so that it'll pinch off and form two daughter cells. So when this is completed, when cytokinesis is completed, the end result is going to be two diploid daughter cells. So the parent cell was diploid. It had 1, 2, 3, 4 chromosomes. This pinches off. We've still got 4 chromosomes. Recall from a previous lecture discussing cytokinesis that formation of a cleavage furrow occurs in animal cells. It does not occur in plants and fungi. Plants and fungi have cell walls. Instead of forming this cleavage furrow, they form what's called a cell plate. The cell plate starts out as a coalescence of vesicles. And these vesicles are derived from the Golgi apparatus. They contain the materials needed to form a cell wall. So cytokinesis in plants and fungi involves the formation of a cell plate via coalescence of vesicles derived from the Golgi apparatus, and the materials in them are used to make a cell wall down the middle of the cell, and that will eventually divide the cell into two daughter cells. Summing up, mitosis, starting out with two sets of 2 chromosomes. I had chromosome 1, chromosome 2. One maternally derived, the other paternally derived. Maternally derived, paternally derived. This cell is therefore 2N or diploid. It's been through S phase, so here are sister chromatids. Each one of these chromosomes has enough information to supply 2 daughter cells. Prophase, metaphase, anaphase, and telophase occurred. And after that, the result is 2 daughter cells. They are identical. They're identical to each other. They carry all the information that the parent cell had, and they are diploid. So that's the most important points of mitosis. It is separation, it is cell division, resulting in 2 identical daughter cells with conservation of chromosome number. All right, to practice, we're going to look at 4 examples. Label the parts of the spindle apparatus below. What is the name of the structure on the centromere to which the spindle fibers attach? Starting out with this first part. Here I have a centrosome. Within it, what's really shown here is the centriole, but the centrioles are part of the centrosome. Remember that the centrosome is a microtubule organizing center, and within each one is 2 centrioles. Same thing on the other side. Radiating out in a star pattern is an aster. So there's an aster on this side and an aster on the other side. Here I have spindle fibers or spindle microtubules. These are composed of microtubules, hence the name. And these are attaching to the sister chromatids. This next part of the question asks me, What's the name of the structure to which the spindle fibers attach? So the centrosome is here in the middle, holding the sister chromatids together. And on the centrosome are two kinetochores. The spindle fibers from one pole attach to one kinetochore. The spindle fibers from the other pole attach to the other kinetochore, giving the setup that will allow the sister chromatids to be pulled apart. So the answer to this question is that the spindle fibers attach to the kinetochore. And the kinetochores are located on the centromere. Second example. Example 2. What is the last phase of mitosis called? What occurs during this phase? So the last phase of mitosis is telophase. Remember, there's PMAT, prophase, metaphase, anaphase, telophase. Some of the events here are actually just a reverse of prophase. So remember, in prophase, the nuclear membrane broke down. Now, the nuclear membrane reforms. And one nuclear membrane will form around one set of chromosomes at one pole of the cell. The other nuclear membrane will form around the other nucleus at the other pole of the cell. So now there's two nuclei. The second event, recall that chromatin condensed in prophase. Now, chromatin becomes less condensed. So once it decondenses, the chromosomes are not going to be visible via light microscopy. During prophase, the spindle apparatus formed. Now, spindle apparatus breaks down. In addition, the nucleoli reappear. Also, you should note that cytokinesis is continuing on. It began in late anaphase. It's also continuing on during telophase, but it's often treated as a separate phase. So for telophase, we're going to put these 4 events as telophase and keep cytokinesis considered as a separate event. Example 3. List 3 events that occur during prophase of mitosis. So prophase of mitosis, remember, we just talked about telophase. That makes this question easy. It's the opposite. Here we're going to have breakdown of the nuclear membrane. The next event is going to be that the chromatin condenses, becomes wound more tightly. Now the chromosomes are going to be visible via light microscopy. The spindle apparatus begins to form in preparation for anaphase, when sister chromatids are going to be separated. So the spindle apparatus begins to form. During this phase, the nucleoli disappear. They are no longer visible. Okay, so prophase is the first phase of mitosis, and these 4 events occur during prophase. What phase, example 4, what phase is the cell below shown in? What events occur during this phase? Looking at this situation here, I see that the sister chromatids are being pulled apart. Since they're being pulled apart, I know that the cell is in anaphase. So the main event of anaphase is separation of sister chromatids. This cell started out with 4 chromosomes with sister chromatids. Now the spindle fibers are going to shorten. They're going to pull this towards each chromatid towards the opposite pole of the cell, resulting in daughter cells that have 1, 2, 3, 4 chromosomes, but only 1 sister chromosome. chromatid. That concludes this lesson on mitosis here at educator.com. Thanks for visiting.